Good afternoon. This is Elad, CEO and co-founder of Astrolabe Diagnostics, and I am joined here today by John Quinn, the Director of Science and Product Development at Bioinformatics. And we are going to tell you about a state-of-the-art analysis workflow for cytometry data, which combines the Astrolabe cytometry platform and FlowJo. And before we begin, if you have any questions, please use the Q&A button below. You can click on it to submit your question, and at the end of the seminar, we're going to try and answer as many of them as we can. So just a brief introduction to me. I've been a computational biologist for over a decade now and one of the first bioinformaticians to work on the CyTOF. I'm the first author on Visney and Wanderlust, and I've been involved with several other methods. And formerly, I've been a staff scientist at the Mount Sinai Human Immune Monitoring Center. All of which to say that I have a lot of experience with analyzing single cell data, but in addition, I'm also experienced with taking these results and delivering them to bench scientists and immunologists. And one of the common stories I encountered over the years is that a researcher is really excited about a given cytometry dataset, whether it's the CyTOF, the Aurora, the Symphony, or just a large dataset with 14 color flow. And they acquire the data, but once they get to the analytics, they get a bit stuck. And there's a plethora of tools out there that can help you analyze your cytometry data, whether it's Visney and FlowSum and the R programming language, and many other tools. So there is a rich ecosystem, but the learning curve for this ecosystem is quite steep. And even once you know how to use these tools, applying them to a new data set is quite time consuming. So from my perspective, what researchers want is some way to take their single cell data and convert it into meaningful biological insights. And what we offer at Astrolabe is an online platform that lets you take care of the mechanics of single cell analysis in just a couple of hours. So Astrolabe is an automated consulting service where you as the researcher can upload your data to the cloud, set up the experiment design the same way you would with a bioinformatician, and then the platform kicks in, takes care of the analysis, and offers you the results through a user-friendly interface. Astrolabe supports flow, cytometry, and CyTOF, human, mouse, many different tissues, many different diseases. And most importantly, we're not some faceless software company. So in addition to the platform itself, we offer what we call researcher partnership, where if you have any questions about the analysis, what does this cluster mean? Can you tell me more about the statistics? Is this real signal or just noise? You can email us and we will help you address it. The platform has many different features, starting with debar coding for CyTOF data, cleaning for both CyTOF and flow cytometry data, a quality control dashboard that covers staining quality and sample quality, automatic cell subset labeling, unsupervised clustering, many different follow-up analyses, including dimensionality reduction, differential abundance, and differential expression analysis, and various export formats, some of which I'm going to cover later in this seminar. So it's quite a lot. Unfortunately, we can't go over all of it today. So I'm going to focus on what I believe is the heart of the research project, which is the follow-up analysis. Really, this step that lets you focus on the high-level ideas and on the biology in your data. In order to demo the platform, I'm going to use this chikungunya dataset that was acquired on the CyTOF by Adib Rahman at the Human Immune Monitoring Center in Mount Sinai as part of the Human Immunophenotyping Consortium. And this is an immune monitoring study where pediatric patients who were sick with chikungunya arrived at the clinic in Nicaragua, and blood samples were taken from them during 2 stages of the infection, during the acute and convalescent stages. And the goal of the study is to compare the structure of the immune system between these 2 time points. What you see here is everything you need to do to work with Astrolabe. You upload your FCS files and you set up the experiment design. In this case, the experiment design is pretty straightforward. There's patients and there's time points. However, this section of the interface is like a spreadsheet. You can have more columns, you can have more values. So for example, you could have an experiment with controls and patients. The patients have 3 different treatments. Each treatment has 4 time points, and then you're comparing responders and non-responders. So you could encode all of that within Astrolabe and then use it later for the statistical analysis. I mentioned that our emphasis is on biologically meaningful insights, and I'm going to show you some of the ways in which Astrolabe helps you with that. I'm going to switch to this visualization called the Cell Subset Navigator. And the entry point to the Cell Subset Navigator is a figure called the MDS map. This is a dimensionality reduction view. For those of you who are familiar, it's very similar to TESS Knee or to FlowJo trees. We have these different bubbles that correspond to cell subsets, and their position on the map corresponds to their relationship in the high dimensional space. In other words, similar subsets like these different T cells are next to each other, while dissimilar subsets like these T cells and B cells are far away. And as you can see here, from the get-go, Astrolabe is speaking the language of immunology. So these different clusters, these different bubbles, have already been automatically labeled by the platform. So there's different myeloids, different NK cells, T cells, and B cells. And when you upload your FCS files to Astrolabe, the platform is using your panel to build a labeling hierarchy for the different cell subsets. You can always access it, so it's over here. For example, in order to label the CD8, the effector memory CD8 T cells, Astrolabe starts from CD3 positive and then CD8, and then looks at 45RA and CD27. So again, you upload your data and you can immediately start thinking about myeloids and B cells and T cells. This view is fully interactive, so I can color code it. For example, here are the CD3 positive bubbles, and I can click on any of these to get more information about the cell subset. So at this point, I would like to fast forward a bit and go back to the research question. So the goal of this experiment is to compare the acute and convalescent time points across the patients in the study. And to do that, Astrolabe offers a tool called Differential Abundance Analysis. I'm going to turn it on for the time point feature. And when I do, 2 things are going to change in the interface. First of all of the bubbles are color-coded based on the fold change between the acute and convalescent time points. In other words, the deeper the color, the greater the fold change. And for example, we can look at these double positive monocytes and see that they're bright red, signifying a high fold change between them. Scrolling down, Astrolabe is offering us a volcano plot where each one of these dots is one of the cell subsets. The x-axis is the fold change between the 2 time points. The y-axis is a measure of statistical significance. It's the negative log 10 of the FDR. The higher it is, the more significant the results. And again, looking at this, I can see that one cell subset is really high on the fold change, and it's the double positive monocytes that we've seen before. If I click on them and scroll back up, I can see them in the MDS map, circled in black. And if I scroll down, I can find more information about this subset. So we're looking at the double positive monocytes, and the first visualization we get are these bar chart, where each of these bars is one of the samples. The y-axis is the frequency of the monocytes in that sample, and they're color-coded based on the time point. So the green ones are acute, the purple ones are convalescent, and we can immediately see how all of the acute samples have a higher frequency than almost all of the convalescent time points. We can get additional statistics here to the right. And in this box plot, the white line is the median frequency of the subset. And we can see how it's 3% for the acute time point, and then it drops by 6-fold into about 0.5% for the convalescent time point. And finally, this is all quantified using the statistics. So Astrolabe is doing everything by the book. This is logistic regression, followed by multiple testing correction. And you can take these figures, you can take these numbers and put them in your next paper or grant application or report, because everything here is backed by the statistics. And in effect, Astrolabe is following the breadcrumbs that we left behind. It's using the panel to find the different cell subsets, and it's using the experiment design to give you this specific intelligence that really focuses on your research question. So I'm going to switch to a different cell subset. I'm going to turn off the monocytes, and I'm going to look at these naive CD8 T cells. So I click on them and scroll down, and we can see that their fold change is high and the FDR is high. So again, there's a statistically significant signal here. And if we go down, we can see that this subset has a frequency of 5% for the acute time point, and then it doubles to 10% in the convalescent time point. If we go further down, Astrolabe is giving us a heat map that allows us to characterize the signature of these naive CD8 T cells across the entire experiment. And this heat map is pretty big, because each one of these rows is one of the samples in the experiment. Each one of the columns is one of the markers. So this is a cytoph panel. It has 30-something markers in it. And we can see that the naive CD8 T cells are positive for CD3 and CD8, and they're negative for CD4 and CD19, all of which trends that we would expect. However, they seem to be heterogeneous for CXCR3 and CD57. And I think that one of the most exciting things about high complexity cytometry is that you can include these additional markers that have biological significance, but are not as well characterized as, let's say, CD14 or CD56. And we want to use them in the analysis. So to do that, Astrolabe is offering a mechanism which is called a second cell subset level. What I'm showing you here is the assignment level, and all of these bubbles correspond to canonical subsets, which we found using rules that come from immunology. The second level is called profiling, and when I click on it, you're going to see that we have many more bubbles. And the reason is that Astrolabe took each one of the subsets and further stratified it using unsupervised clustering. So these still correspond to classical monocytes, but we have many more of them, again, because they've been broken down using clustering. This step is completely agnostic. At this point, Astrolabe doesn't know anything about biology. All it has is the signal that comes from your data. And specifically, the naive CD8 T cells have been broken down into 4 clusters over here, highlighted in black. So as a reminder, before we saw a twofold change when examining the entire naive CD8 T cell compartment. When we look at the volcano plot now, we see that, that one of these clusters actually has a much higher fold change than the others. And if we focus just on this cluster and scroll down, we see a 10-fold change in frequency between the acute and convalescent time point. So Astrolabe has taken us through a journey where we gave it the data, we explained the experiment design, the same way we would interact with a bioinformatician. Astrolabe takes care of the cleaning the data and labeling and clustering and the statistics, and gives us very specific intelligence. Namely, within the naive CD8 T cell compartment, there's a specific cluster, and that cluster changes significantly in frequency between the 2 time points that we defined. So this was a brief taste of the capabilities of the platform, and as I mentioned previously, we actually offer a plethora of different features that you could use for your research. And just to give a brief summary of the main ideas behind Astrolabe, first of all, it is very easy to use, and you don't need to know R or programming to access all of these powerful tools. You just need to use the web interface. The second part is that Astrolabe analysis is extremely fast. So we can go through 10 or 100 or even 1,000 FCS files in just a few hours. That means taking the files and cleaning them and labeling them and clustering and so on. And for you as a researcher, you can acquire the data, put it in Astrolabe, go grab lunch, and then come back and start thinking about these high-level questions that come from your data set. And finally, all of the Astrolabe results are open to follow-up analyses. You can use the R programming language for that. We have spreadsheets that you can import into Excel. We have the online interface, of course. And finally, all of the results are available through FCS files, which you can import into FlowJo. So the next part of the seminar is going to focus on FlowJo and how you can use a combination of FlowJo and Astrolabe to really dig deeper into your data in a very efficient way. So let's talk about getting the Astrolabe data into FlowJo. And the starting point for that is within Astrolabe, you have all these different buttons, and these let you download the Astrolabe analysis in various formats, such as R files, such as spreadsheets and PDFs, and finally, these gated FCS files. And when you click on this, you're going to get a zip file, and that zip file has all the FCS files in the experiment, except they have the Astrolabe ending, and they have additional columns for the assignment and profiling level. So we're going to put these into FlowJo, and I'm not going to put all of them just because putting all of them is going to take a bit too much time for this seminar. So I'm going to take 5 patients and drag and drop them into FlowJo. So before I continue with FlowJo, I will say 2 things. One, I think every person has a bit of a different style on how to use the software. So whatever I'm doing, you might be doing it differently, and it can definitely be done in many different ways. And the second thing, when we just started working on this webinar, John and I, my idea was that I'll present the Astrolabe part and John will present the FlowJo part. And then John said, well, the FlowJo part is pretty easy, so I want you to do it. And then we had like a 20-minute conversation on what I want to achieve with FlowJo, and I really got a hold of the software really quickly. So I was pretty impressed with how friendly and accessible FlowJo was. So we have all of these different samples, and we can click on them to see the biaxial plots. I just want to tidy them up a bit before I continue working. And to do that, similarly to the features from Astrolabe, I'm going to add these keywords. So I'm going to add 2 keywords, one for patient and the other for time point. And there's different ways to populate them. You can use Excel to populate them. In this case, there aren't that many samples. Sorry about that. There aren't that many samples, so I'm just going to type the sample IDs, and that's going to take just a few seconds. But again, you can just import this from an Excel spreadsheet. So if you have more than 10 samples, you probably want to do that. And the second thing I'm going to do is I'm going to use this feature called Create Keyword Value Series to add the time points. And to do that, I'm going to tell FlowJo that I have 2 samples, and they're going to be called acute and convalescent. And once I do that, this is all replicated here. And now, again, just to make things a bit more comfortable, I'm going to change the way we refer to samples. And instead of referring to them via the file names, we're going to refer to them via the patient and the time point. Okay, so now it's much more easier to look at the data. I'm going to save this workspace. And we are going to call it demo. Let's put it here in FlowJo webinar. And now we can start looking at the data. So I mentioned that Astrolabe stores the assignment and profiling level in the FCS files, and this is through new columns which are called assignment and profiling. So I'm going to turn to the assignment column, and again I'm going to tidy it up a bit. I'm going to turn it into linear axis, and I'm going to customize it so that it goes up to 45, let's say. So now each of these peaks, each of these streaks that we see here are one of the subsets that Astrolabe found. For example, if we put CD3 on the y-axis, we can see that some of these streaks are higher for CD3 and some are low. So some are for the T cells and some are not. So unfortunately, FCS files cannot store strings. They can only store numbers. And we need some way to take these numbers and convert them into the actual labels. And Astrolabe provides us with a spreadsheet called assignment. And if we click on it, it's going to be a map from each one of the values to each one of the cell subsets. So one thing we could do is we can switch to a histogram view. and then right-click and use Create Gates on Peaks to break down each of these peaks into the respective population and then label them manually. Thankfully, we don't have to live that way, and FlowJo offers a plugin called Astrolabe Import that lets you import the Astrolabe labels straight from the CSV file. So you can get this on FlowJo Exchange. And over here, I'm going to go to Plugins and Astrolabe Import, and I'm going to choose the assignment CSV. So it's going to ask me how do I want to prefix the subset names. I'm just going to delete this for now. And now we can see all the different subsets. So for example, if I plot CD3 versus CD19, and then switch into the CD27 B cells, we can see just the B cell population. And if we switch into the naive CD4 T cells, we see just the T cells. So we can take this Astrolabe import and drag it into all samples. And once I do that, all of the samples in the experiment are going to have the Astrolabe subsets in them. So far, so good. And as I mentioned, we can use this view to see different biaxial maps and compare the various cell subsets and see how they look. However, I think that it could be beneficial to validate the data in a bit more of a effective way. And to do that, FlowJo offers the layout editor. And what the layout editor allows you to do, it allows you to generate a figure on one of the samples, and then you can use this batch feature to replicate it over all of the other samples. So what I'm going to do with the layout editor, I'm going to start with the B cells and drag them here. And now we're seeing the biaxial plot we had before, the CD3 versus CD19. And I'm going to drag it again, and we have the same figure again, but I'm going to change the y-axis, the x-axis here from CD3 to CD14. So now we have 3 of the major markers, CD3, CD19, and CD14, and I'm going to grab a bunch of other files. So I'm going to grab all the other subsets. Sorry, let's make it a bit more clear. Let's just grab the major ones. So let's grab the naive, and the classical monocytes. And I'm going to drag them here as well. And once I do that, the color scheme changes from density into this legend that shows us the different cell subsets. And specifically, the myeloids are in orange, the T cells are in red and in blue, and the B cells are in green. And we can use these 3 biaxial plots to validate that the Astrolabe assignments are correct. The T cells are positive for CD3, the B cells are positive for CD19, the myeloids are positive for CD14. So I'm going to make this a bit nicer to look at by changing the fill into black. So it's going to be easier to see the different subsets with a black background. And just so we can keep track of the various samples, I'm going to add a text box here. And the text box is going to have the patient and the time point. So for the patient, we're going to insert the patient keyword. And for the time point, we are going to insert the time point keyword. And now we have this nice visualization that allows us to compare the different subsets that Astrolabe found for this sample. And now we want to expand it into the other sample. So I'm going to go to the layout editor, instruct it to have just one column. So every sample is going to have its own row. And I'm going to create batch reports. And once I do that, you will see that FlowJo now replicated the 2 figures I started from and created a version for each one of the samples in the experiment. And we can use this text box to see what they are. So this is 1758 acute, convalescent, 760 acute, convalescent, etc. And we can zoom out to have a quick look and validate the Astrolabe results, and indeed see that all of the cell subsets were or assigned correctly. So this is a really cool report for, again, assessing the quality of the subset labeling and learning a bit more about your data. But we would like to take this a step further. And one of the powerful algorithms that are very popular for analyzing high complexity cytometry data is TESS Knee. TESS Knee is a dimensional reduction algorithm that takes all of the cells and puts them on one biaxial plot. And the positioning of the cells in this single biaxial plot uses information from all of the channels. So I'm going to close the layout editor and run TESS Knee. So FlowJo supports TESS Knee, and in order to use it, we first need to concatenate all of the samples into one FCS file. So to do that, I'm going to go to Edit and Collapse All. So I just see the FCS files. I'm going to choose all of the FCS files, and then right-click on them and click on Export Concatenate Populations. So this is going to allow me to concatenate all of the files into a single FCS file, and it's going to put the file here. I'm actually going to move it to the correct directory. Here it is. And if I click on Advanced Options here, then I have the option to add additional parameters. For example, I can add the patient and time point parameters. So let's find them here. Here's patient and here's time point. And I can set a custom set of parameters. What I'm going to do is I'm only going to take the parameters I need for TESS Knee and for the follow-up analysis. So I do not need time. I do not need some of these empty channels. I don't need viability or the D-bar coding channels, and so on and so forth. So this is going to make the file a bit slimmer, and it's going to make my follow-up analysis a bit easier. So I pick just the files I want, and then I'm going to click on Concatenate. And now FlowJo is concatenating all of the files, and I'm going to ask it to put it in the existing workspace so that I can continue working on it. So concatenation is done, and the file is here. And I will point out that it has a bit over 1 million events. And it also already has all of the subset labels from Astrolabe. So now to actually run TESS Knee, I'm going to go into Workspace and click on TESS Knee. And that's going to open the TESS Knee dialog for FlowJo. And there's a few parameters we need to choose when running TESS Knee. The first one is which markers we want. And thankfully, that's going to be relatively easy. We can just click on them here and pick these markers. Don't include any of the Astrolabe channels, because that's going to make TESS Knee's job a bit too easy. So we're going to pick all of these. And then TESS Knee has various parameters, such as iterations, perplexity, and learning rate, and so on. Thankfully, earlier this year, Annabelle Kina et al. Released a variant of TESS Knee called OpsKnee. And FlowJo implemented OpsKnee. OpsKnee chooses all of these parameters for you. So there's no more need to deal with the different parameters on TESS Knee. You can just use OpsKnee, which is what I recommend right now. Additionally, you need to choose these two algorithms called KNN algorithm and gradient algorithm. And each of these has two variants. One is more accurate and one is faster. In the case of TESS Knee, the loss of accuracy is really not an issue. You can choose the approximate algorithms for both of these and still get excellent results. In fact, I tried using both exacts and this approximate version, and both of them made perfect sense, the TESS Knee maps. So you do this and you click Run, and TESS Knee is going to start running. I'm not going to show it here because it does take a few minutes. However, I want to point out that it doesn't take that long. So there's 1 million events here, and running TESS Knee on these 1 million events using FlowJo takes about 15 minutes. So pretty boring for a seminar, but definitely something you can do on your laptop or on your desktop computer at the lab. So I'm not going to click on this. Instead, I'm going to close the TESS Knee dialog, and I'm going to open a TESS Knee run, which I did before the webinar. So this is how the results of the TESS Knee run looks. And I can click on this to open the biaxial plots, and within the biaxial plots, I can look at TESS Knee 1 and TESS Knee 2 to see the results of the TESS Knee analysis. So here's the TESS Knee map, and we don't know what any of these cells mean yet, but we will be able to in a minute. Before we do that, I want to split the concatenated FCS file back into their respective original files. So to do that, we can pick the sample ID channel, and when FlowJo concatenates the files, it creates this channel so we can go back to the files from which we started. So we're going to do this and choose histogram, and then right-click and create gates on peaks. And once I do that, FlowJo takes each one of the peaks in this channel and creates a new file from it. And you will see that these correspond to the files that we started from. I can click on them, right-click, and then export them, and use the export dialog to get back the original files from which I started, except now they're going to have the TESS Knee channels. And then once I have them, I can import them into FlowJo, set up the experiment, add the Astrolabe import plugin, and continue working on it. So I am going to jump ahead a bit and show you how the file analysis looks like. So what we have here are all of the various FCS files, and for each one we have the TESS Knee channels, and for each one we have the patient and the time point. And just by looking at this, we can start looking at the TESS Knee map and see what changes. So here is the TESS Knee map for one of the acute patients, and if I click here, now this is the TESS Knee map for the same patient, but the convalescent. And we can already see how quite a few densities change. Notably, look at this big blob here. This is the left side is in the convalescent, and the right side is in the acute. So we would like to investigate this a bit more closely. And once again, we can use the layout editor. So going over the same steps I did with the biaxial plots, we can do the same thing for the TESS Knee channels. And what I'm showing you here is a big report that started from this one figure. This one figure for one of the samples. It has the TESS Knee axes. It has, again, the text box with the patient ID and the time point. And I color-coded the points based on the different subsets that we got from Astrolabe. And when we do that, we can compare each acute patient to the left with each convalescent patient to the right. So again, acute, convalescent, and so on. And furthermore, I have set up the heat map, the color coding, so that monocytes are shades of red, CD8 T cells are shades of green, CD4 T cells are shades of blue, and B cells are shade of pink. And we can see that the big blob that we saw transition before is a group of monocytes. And it looks like the monocyte composition changes quite a bit between the acute and convalescent time points. So with traditional gating, we would see just one uniform set of monocytes, and TESS Knee really helps us better identify the specifics. Another thing that we can see here is the change in the double positive monocytes. They're in dark red, and we can see how they go down between the acute and convalescent. And finally, we can start thinking about new things. So there's this light pink blob to the top left that seems to reappear in the convalescent time point. So we barely see it in acute, and it reappears in convalescent, and this seems to be consistent across all of the patients. So here's the last patient. Again, there's some of them in the acute, but many more in the convalescent. If we go back to the legend, these seem to be plasmablast B cells. So we notice something in the TESS Knee map. How can we follow up on it? Well, how about we go back to Astrolabe? So let's go back to the Astrolabe view, go back to the assignment level, and find the plasmablasts. So the plasmablasts are down here. And if we scroll down, we see that there is some trend there. Not as strong as the double positive monocytes, not as strong as the naive CD8 T cells, but the trend exists. And Astrolabe helps us quantify it. Astrolabe helps us get the frequencies on a sample-by-sample basis. We see that there is about a 50% increase in the frequency of these subsets between the acute and convalescent time point, and we have the FDR score to quantify it. So the workflow is very organic. We start with Astrolabe, we give it the data, we give it the experiment design, we start generating high-level insights, and then we can seamlessly transition into FlowJo, generate validation plots, run TESS Knee analysis, and use this TESS Knee analysis both to follow up on the ideas we got from Astrolabe and generate new ones. And then we can take these new ideas and again go into Astrolabe and see whether we can quantify them in a more rigorous manner. So once again, I am going to thank John Quinn and the rest of the FlowJo team. And I'm going to thank you as the audience for listening to this webinar. And now we're going to take a few seconds break and address some of the questions that we got in the Q&A. So just one last reminder, if you have any last-minute questions that you want to post, please use the Q&A button below. And we won't be able to address all of them, but we are going to answer as many as we can. So we have a bunch of questions in the Q&A, but before I go there, it just occurred to me that I didn't let John say anything. So hey, John, welcome. John, are you there?
How about now? Can you hear me?
Yes, so welcome. And would you like to spend a minute to introduce yourself?
Sure. Hi. And for those of you who don't know me, and I see a few familiar names on the list, I'm John Quinn, the Director of Science and Product Development for FlowJo. And so, including teaching how to use FlowJo, it's also my job to help further decide what features should be included and where the program should go. And so it is fair to write me at the email address listed there, john.flowjo@bd.com, with any requests for inclusions or anything that we're not doing well enough. And that's, I believe, a good enough introduction for me. I am the FlowJo guy, and that's probably just enough.
Yeah, thank you. And as I mentioned earlier in the webinar, I managed to get very far in FlowJo very quickly thanks to your assistance. So thank you for that. So we have a few questions. The first one is, are we going to send the video of the webinar? And yes, we will. Once the webinar is done, we're going to email everyone with a link to the webinar itself. And going into the science a bit, one question is, can you clarify if you concatenate the previously identified subpopulation for the TESS Knee? For that, so we concatenate all of the FCS files, and the FCS files have the Astrolabe assignment and profiling columns in them, so they're concatenated as well. Which means that after you run TESS Knee, you look at the TESS Knee map, you can go back to the Astrolabe labels and show them on the TESS Knee map. Next question is, why not doing an unsupervised TESS Knee? So to clarify, the TESS Knee is unsupervised. The TESS Knee is using all of the cells. The TESS Knee doesn't know anything about the Astrolabe labels, and we just show the labels after the TESS Knee is done. So these are two separate steps. They don't know anything about each other. Next question is, why did you not downsample each sample to the same amount of cells? You can definitely do that. I will point out that the statistical analysis within Astrolabe does factor the number of cells in a given sample. And furthermore, within FlowJo, when you export, you can downsample. When you concatenate, you can downsample. I have a question, is Astrolabe import already available in FlowJo? Yes, it is. And I can actually show you, if you go into the FlowJo Exchange, then one of the plugins there is going to be the Astrolabe import plugin. So another question I have here is, I think this one is for John. Could you please elaborate about the TESS Knee approximate algorithms and OPSNI? So I'm guessing the reference is to this part. And John, could you elaborate a bit about that?
Yeah, absolutely. There's a couple different choices now. OPSNI, as mentioned by Alada, is an automated way to set most of the parameters that can determine how that the algorithm is going to run. And prior to having OPSNI, you would basically have to guess at what point. Well, TESS Knee effectively has two portions, one where you try to agglomerate similar things, and a second portion where you try to push apart things that are dissimilar. In setting just an eta value by a guess and a perplexity by a guess and so forth, you're just hoping it works out. If you use OPSNI, then the algorithm can actually calculate how the goodness measure is doing and switch portions from pushing to pulling together at the appropriate time when you've hit a nice plateau and you've realized you've gotten the most you can out of each particular portion. The other two algorithms at the bottom, KNN and the gradient descent, the first part of TESS Knee, because what TESS Knee is attempting to do is figure out which cells are near each other at the high dimensional level and represent them on a plot where they're still near each other in a low dimensional representation, is calculate which cells are near each other by distance, by figuring out which cells are neighbors. And so we can start out by either calculating the distance between every pair of cells, which is extremely time intensive, but it will be exact, or we can actually steal an algorithm that Spotify is using to determine which music to play next for you, which is to make essentially the assumption that if there's two cells and there's a third cell that's a neighbor to both of them, then the neighbor of your neighbor is your neighbor. And if the other cell is not a neighbor of your neighbor, then it's not. And we can reduce the number of calculations we go as we move through the data, coming up with a map of which cells are neighbors to each other. And as Elad mentioned, we've done a lot of just test cases to see what you lose by approximating, and it seems inconsequential. I get what appears to be the exact same results using both ways, and the Annoy algorithm seems to be significantly faster. So I really do like using that. The other is a gradient descent. And so TESS Knee is an iteration to the next. The rate at which you tune it can be calculated a number of ways. Barnes Hut, which is the default, has always been the traditional. The fast Fourier transform uses some heavy math.
So John, I think we lost you for a second there.
Faster and slower when appropriate, and I will kind of trust that it goes a little bit faster. Same answers.
Okay, so I'm afraid we lost John at the end of the answer there. But as he was saying,
I'm sorry, did I cut off there?
We lost you when you reached Barnes Hut. So could you please continue from there?
Oh, sure, no problem. Sorry, I'm having an unstable internet day apparently. Yeah, so Barnes Hut has always been the traditional gradation off of the first proposed method. Anyway, it moves from one step to the next throughout the iterations, trying to make the algorithm fit a little bit better. The fast Fourier transformation is just a speedier gradient descent tool to accelerate to larger transitions when needed and smaller transitions when that is more appropriate.
Cool, and I have to admit that TESS Knee has run blazingly fast for me when using FlowJo. So I'm just using a standard desktop computer, and running these over 1 million cells took about 15 minutes. And speaking to you, that seems to be a pretty common experience. Okay, so we might have lost John again. I will move on to the next question. And that is, how does Astrolabe assess the quality of a sample, especially when run in different disease stages and over several time points? And that's a great point. We have many experiments that, let's say, have PBMCs and tumor samples side by side. And evidently, the tumor quality is much lower due to degradation, due to digestion. So Astrolabe has a quality control dashboard where you can see various scores. And some of these are sample specific. For example, how many of the events are debris, how many are unassigned, whether there's been any normalization issues with the beads. And in addition, we have a dashboard for standing quality. And this is using a metric called the average overlap frequency. And this one is a score between 0 and 1. The lower it is, the higher the standing quality. And you can use either one of these dashboards to get a quick look at your data and verify that the standing quality is high. With that said, I can confirm, at least based on my anecdotal evidence, that you can expect a much higher debris rate and a much higher doublet rate in tumor samples and tissue samples. So that is something to be aware of. So John, are you back by any chance, or are we still out?
I'm back.
Okay, great. So one more questions I have here is, can you please elaborate on FlowJo visualizations other than biaxial plots?
Sure. A lot of the FlowJo, like the niftier FlowJo visualizations, are actually built in through a series of plugins. And so biaxial plots can actually house more than 2 dimensions if we use something like TESS Knee. And so there's a few other options that are available as plugins. So we can do UMAP. So UMAP is very similar to TESS Knee, with the difference in that we attempt to scale the data in that case so that the differences or the distances between a set of clusters is perhaps relative to actually how different the populations are. Whereas TESS Knee is just a pure visualization that is going to just pull apart the most different things as possible and just represent them on a plot. UMAP actually has some meaning to the scaling. So there's that. There's also EmbedSum, which is based also on the FlowSum algorithm. It takes any kind of FlowSum results and then tries to produce a dimensionality reduction based on those clustering results. And what's really nifty about EmbedSum is that it's forward extensible. So whereas we did the step here where we concatenated all the data together so that we could do direct comparison of the TESS Knee parameters, FlowSum allows you to preserve the first stage of the clustering algorithm in it, the self-organizing map where the SOM comes for. And so because FlowSum is forward extensible, we can preserve that model, and we can also make EmbedSum forward extensible so that you don't have to have done everything at one time. If you come back to an experiment later in the future and you want to create the same kind of visualization, you can do so. The downside of EmbedSum, as the graphic that comes with the model implies, is that it doesn't produce quite as nice a result as TESS Knee, and that it's got some limitations to it. But it is definitely an alternative. And speaking of FlowSum, it itself produces a minimal spanning tree as a way to represent the populations in the data and the relationship to each other, and that, as the graphic there indicates, each different circle there is a node. The ones that are colored are considered to be similar to each other, and the lines connecting them are distances or relative differences between those clusters. So there's a bunch of different visualizations there. And then some of the others that are just built right into FlowJo. So Elad showed a TESS Knee plot, and he also showed overlaying in the color. There's a few other things that you can do relatively quickly with any of those plots. If you right-click on them, there's a choice for multi-graph overlays, where I think those are yeah, there you go. Those are already grouped together since they were batched. There's a make multi-graph overlay choice there at the bottom, where you can show the data as histograms, all parameters by Y, and you can also break out, if you don't have an overlay of many things all combined, there'll be a fourth choice which just says make a heat map of any parameters that you're interested in. So if you want to explore a data set, you could say create the TESS Knee plot, show X and Y TESS Knee dimensions, and then heat map every other color, if every other color is interesting, across them in a series of plots to get a feel for what each of those islands represent just in one click. I think those are the newest and most interesting representations. Of course, there's 100 different ways that are very conventional to represent data in FlowJo, whether you're talking about histograms, whether you're talking about contour plots, etc. But I won't bore the audience with the description of those.
Yeah, and one of the questions I often get is, how is Astrolabe different from FlowJo? And the answer I like giving is that FlowJo is like the Swiss Army knife of single-cell data. And it has many different tools and many different components, and that's even before factoring in the pretty rich FlowJo Exchange and all the plugins. So if you want to really deeply explore your data, FlowJo is a very powerful tool for doing that, while Astrolabe is more about taking a step back and starting from the high level view of the data. And I think this is why the two tools complement each other so well.
I'd agree. I feel like there's a very nice workflow here that we can put together the two tools and super quickly come up with biologically relevant names that are easy to understand and show the differences between them and get down to the real science of understanding what's happening between two biological systems and spend less time fooling around with hands-on clicking and gating and so forth.
Yeah, I agree. So I think I'm going to answer one last question. Can you combine the results of two flow panels and connect the samples by their shared surface marker? For example, CD19 positive B cells, one in a B cell subset panel, the other one in a whole blood panel. So the way you would do this is you would set up two separate experiments in Astrolabe, one for each one of the tubes, run the analysis, and then probably use either one of our exports. So we have exports that include cell subset frequencies in spreadsheet form or in R files to combine the results, or alternatively, you could go back to FlowJo. And in FlowJo, you can definitely import the FCS files from each of these experiments and compare any results there and generate any overlapping figures there. So I think that covers all the answer we got. John, anything you would like to add?
No, other than to say, thank you for making a really great presentation, for making all this look really easy to do. I appreciate it.
Yeah, it was really easy to do. So thank you. And thank you for the audience for joining us. And I will be sending a video of the seminar later today. Of course, you're welcome to share it with your colleagues and other researchers. So thank you, everyone, and have a good weekend.